Hi, we are the promoters, Shannon, Abdur, Casey, and Dakota, and our presentation is on genetic cloning. First, I will cover some basic history of gene cloning. A definition of a clone is an organism or cell which is genetically identical to the parent organism or cell. The idea of gene cloning dates all the way back to the Middle Ages. Plants were first cloned in the 1950s. Other species of plants followed shortly after. First animals to ever be cloned were frogs in early 1962 by John Gurdon of Oxford. In 1963, it was the first time the word clone was ever used in a speech. In 1969, isolation of the first gene by James Shapiro and Jonathan Bitchwith. In 1972, first recombinant DNA molecules. In 1980, the cloning of lab animals and livestock became popular using embryonic cell techniques. In 1983 was the invention of PCR. In 1987, Dolly's sheep was the first organism with a vertebrate to be cloned. In 1990, the Human Genome Project began. This was an international research program with the purpose of mapping and understanding all of the genes of human beings. Scientists wanted to find an accurate sequence of the 3 billion DNA base pairs that make up the human genome. All of our genes together are known as our genome. Began in 1990 and ended in 2003. Also used for the mouse and the fruit fly. In 2000, we cloned pigs. There was a litter of 5 pigs, and the important part was that pig parts could potentially be used in human transplants, making organs easier to come by. In 2003, Dolly died. She was put down. She suffered from lung cancer, but she had 3 babies in her lifetime. Many advances in genetic cloning have been halted due to ethical issues. The possibility of human cloning has resulted in many countries discarding cloning research.
Genetic cloning begins with the choice of cloning vectors and host organisms. Replicons are capable of replicating autonomously. An example are plasmids, which replicate independently of chromosomal DNA within the cell. Host organisms include E. coli, which are widely available and offer rapid growth of organisms with minimal equipment. Corynebacterium are commercially produced production of amino acids and widely used to produce glutamate and lysine. Yeast is also another example. Preparation of vector DNA include cloning vector treated with restriction endonuclease and to cleave at the DNA site, the sticky end, where foreign DNA will be inserted. And DNA to be cloned are extracted from organisms of interest. DNA is purified to remove contaminating protein, RNA, and smaller molecules. Purified DNA is treated with restriction enzymes to regenerate fragmented ends. Recombinant DNA. As shown in the image below, the sticky ends are linked. DNA prepared from vector and interest gene are mixed at appropriate concentration, and DNA ligase linked the ends covalently.
Now we are moving to introduction of the recombinant plasmid into host organism. So basically this can be done either by transformation, transduction, transfection, or electroporation. Transformation is when the recombinant plasmid is taken up through cell membrane where bacteria is in competent state.
And then the bacteria is grown on the culture dish and is allowed to grow into colonies, and this colony is known as gene libraries. Gene library is screened to determine which bacterial colony is making copy of desired gene. Screening can be made either by detecting DNA sequence of the clone genes or detecting the protein that genes encode or use of linked DNA markers.
This is the summary for gene cloning process. In the very beginning, plasmid will be taken away from the bacterial cell and serve as a vector. As you can see in the plasmid, there are antibiotic resistant gene, LAGZ gene, and multiple cloning sites. The interest gene will also be taken away from the cell. Restriction enzyme called restriction endonuclease is used to break the DNA apart, leaving the sticky end. The sticky end of restriction of DNA fragment will attach to each other.
The plasmid then mix up with bacteria using transformation method, and the antibiotic are added, which then allow plasmid only with antibiotic resistant genes to survive. Bacteria then sorted out, and the bacteria only containing gene of interest is isolated. Then the bacteria is allowed to reproduce.
This is the example of gene which had been cloned, and as you can see, the blue dot represent the bacteria with a LAGZ gene, and the white one is the one of the desired gene.
This research eventually could be used on human hosts, most likely embryos. which makes the ethics of this research very controversial. This is all hypothetical and in the future, but America will most likely be one of the countries that either outlaw this technique or use this technique to save human lives. This research can be used in selfish ways, meaning a wealthy set of parents could go to a clinic and if they are short, they could have their child be tall, or they could give their child the endurance of Lance Armstrong. But that being said, this technique can be used to save lives as well. Cystic fibrosis is a genetic disorder that could be fixed using this method, as well as asthma, diabetes, and infertility. On a very exaggerated note, this technique could potentially be militarized and be used to create super soldiers. The real-world applications of this research include insulin, clotting factors, human growth hormone, cytokines, and anti-cancer drugs. Before the advent of gene cloning, these proteins had to be purified from their natural tissue sources. For example, insulin was purified in cow and pig pancreases. Due to the slightly different amino acid sequence of human insulin compared to pig or cow insulin, there tended to be an immune response in some patients. Gene cloning is also used to produce the molecular tools we use to study genes. Restriction enzymes, DNA polymerase, DNA ligase, and other enzymes are all cloned in this way. This research is also used in forensic science and transgenic plants that produce their own pesticides.